Welcome to General Anatomy and Physiology Online. This is the lecture on the cell and cell transportation. In today's agenda, we're going to review the plasma membrane and the structure of it, the cellular extensions found on some of these cells, the organelles that we find in some of the cells, and then also the mechanisms of cellular transportation, how things move from one side of the cell towards the other side. First of all, we have to understand the cell is the smallest unit capable of life. We have about 60 trillion cells in our body, and they all have a different function within it, and a lot of them are shaped differently. Structurally, they are shaped differently. Some of them have nucleus, some do not. Some have certain organelles, some do not. Some have certain extensions, some do not. So their structure and their function are different amongst one another. And so the first thing that we'll look at here with the cell is the plasma membrane. And so the plasma membrane, if we go back here, this is the barrier, if you will, between the inside of the cell and the outside of your cell. And it is very thin. It is semi-permeable, which means it only allows certain things to move back and forth between it. It's very flexible and elastic, and so how I would want you to imagine a plasma membrane would be like if you were blowing a soap bubble in the air, and the soap bubble would be moving through the air. It would be moving and be flexible, but yet very thin and fragile at the same time. In a plasma membrane, our cells plasma membrane of our cells are very similar to that. And its job is to divide this intracellular fluid from the extracellular fluid and keep that as a barrier and allow it to be somewhat semi-permeable, which will allow nutrients to go in and waste products to go out. But the idea is to make sure that the volume of the water on the inside stays the same. And what goes in and what goes out as far as that goes in volume, because obviously if we have too much fluid inside the cell, it could burst. If we have too less fluid, it could dehydrate, and shrivel up, if you will. So we want to maintain homeostasis here. The components of the plasma membrane are the phospholipid portion of it, and if you look here, you'll see the sandwich effect. It's called the phospholipid bilayer, bi meaning two. This is one layer right here, and you can see the other layer here. And what we're looking at here are these round little heads with these two little tails that are pointing inward. And this would be one layer here and another layer here, and it's like two pieces of bread. And we'll talk about this in a little bit, but there's one section here that is more water-loving, which is the phosphorus head here. And notice that it's pointing inward towards the water and outward towards the water here. And the phosphorus portion, the little round section here, this is what we call hydrophilic, if you will. Hydrophilic, which is water-loving. The little fatty acid tails right here that are pointing inward are away from the water edges, so these would be hydrophobic, they're water-fearing. The other components would be these large purple structures. Some of them go all the way through, some halfway, some have little channels. These are the proteins within the plasma membrane, and we'll be introducing you to those as we move along in this unit. Or in other units, excuse me. Next we have carbohydrates, which are always sticking on the outside of the cell here in green, and they help guide cellular reactions and define your cells of what that they're your cells and not somebody else's cells. And you'll hear me talk a little bit more about that in the lab. And then within the plasma membrane are the cholesterol, which kind of helps keep the plasma membrane rigid and as strong as possible. Okay, and we just kind of covered this on the last page, so you can ignore that for the most part. Here are the components that we just described on the last page, and be able to recognize them if you did see them. And next we move into the extensions of the plasma membrane. And so we have three major extensions. We have the flagella, the cilia, and the microvilli. And so the first one is the flagella, and this is the only picture that I have of the flagella. This is a cell right here, and it has one extension coming off of it. This one extension is the flagella, sometimes called the flagellum. It is more commonly seen on a sperm cell. Next, we have cilia and microvilli, and the first one is cilia. Cilia here, these would be your cells, and you can see these extensions coming off of the cell. Cilia are very flexible. As you can see here, they're bending in different directions. They are hair-like, and their purpose is to move substances across the cell, as you can see here. Where we commonly see cells that have cilia would be in the respiratory tract. As you can see, especially here, I'm describing the mucus that would cover the cells on top, and so the cilia would help move the mucus across the cell. Microvilli look like maybe they could be cilia, but notice they are not flexible. They're very hard and rigid. They stick straight up. They have very little to no movement at all. These are going to increase surface area, and a good place to have surface area would be where you are absorbing materials, such as the intestinal tract. We want as much surface area to absorb as much nutrients as possible. This is a good place for that. The cytoplasm, also known as the cytosol or the intracellular fluid, is the liquid that we find within the cell. It is also would include the substances that would be suspended within the liquid, such as what we call inclusions. And so inclusions can be many different things: amino acids, sugars as examples, but these are things, inclusions are things that would be suspended within this liquid. And so the cytoplasm is not just the liquid, but it's also these other things that we find dissolved or suspended within it. Centrioles. Centrioles, there are a pair of them that sit perpendicular to one another, as you see here in your cell, and they are tube-like, as you can see here. And their purpose is to help with mitosis or division of the cell. And so these little tubes, as you can see here, they start to expand, and basically they help with cell division, a little bit more specifically with guiding the chromosomes to one side of the cell or the other. And you'll learn more about that in lab. The mitochondria is known as the powerhouse of the cell. We have many mitochondria within our cell, and they're known as the powerhouse because their job is to produce lots of ATP, which you will learn more about ATP in our metabolism lecture here coming up. And they play a pivotal role in helping generate lots of ATP within your body. Ribosomes. Ribosomes are made in the nucleolus, which is, I'm going to go to the next page here, which is the dense center of the nucleus. Okay, so this whole thing right here is the nucleus, and in the center here is the nucleolus. This is where ribosomes are manufactured. Ribosomes then leave, and they're found in two places within our body, or our cells. They can be floating in the cytoplasm, okay, or they can be attached to another structure which we're going to talk about in just a moment, called the endoplasmic reticulum rough. And the purpose of the ribosomes is to help with protein production, which you will have another lecture in this unit coming up on protein production. So this is a very important piece to the protein production process. Here's our endoplasmic reticulum. This one is the endoplasmic reticulum rough. It is attached to the nucleus membrane, if you will, as you see here, the nuclear membrane. And you have endoplasmic reticulum rough, which has all these ribosomes on it. And you also have an endoplasmic reticulum smooth. And so the purpose of the endoplasmic reticulum rough is to help with protein production. Because it has the ribosomes on it, as we know, ribosomes help with protein production. So if the endoplasmic reticulum has the ribosomes attached, its role too is to help produce protein. On the other end, we have the smooth endoplasmic reticulum, and it does not have ribosomes, so its job is not to produce ribosomes. It has several different purposes. I'll give you a few of them. One is to produce cholesterol, so our liver cells have a lot of this smooth endoplasmic reticulum to produce cholesterol, which is normal for our body to produce. Also, it detoxifies your cells of any excessive narcotics, whether they be prescribed or not, alcohol, anything that it needs to kind of get rid of, any waste products within the cell, the smooth endoplasmic reticulum can help with that. Our next structure is the Golgi apparatus, and the Golgi apparatus kind of looks like it could be an endoplasmic reticulum of some sort, but it's not attached to the plasma membrane at all, or excuse me, the nuclear membrane. It's kind of floating out on its own. It looks like stacks of pancakes, as you can see here. And what happens is there's a transport vesicle that comes from the rough ER that's filled with proteins, and it drops the proteins off in the Golgi apparatus. And basically what the Golgi apparatus does to these proteins is it modifies them. It might add some sugar to it, it might change them a little bit, but then at the end it packages them up into vesicles, as you can see here. So we always know it's a Golgi apparatus when we see a lot of vesicles kind of floating around it. And so we sometimes call the Golgi apparatus the packaging center, because this is where it packages up the finalized protein, if you will, within our body. So there is a relationship between the endoplasmic reticulum rough and the Golgi apparatus, as you can see. You know, the endoplasmic reticulum rough over here produces the proteins, and those proteins then move over into the Golgi apparatus where they get modified and they get packaged finally. Some of those proteins might stay within the cell, as you can see right here, and some of them get shipped outside of the cell to be used within our body or to be used by other cells. And in this picture here, you can see this is the endoplasmic reticulum rough. These are the little ribosomes here, and what they're creating here is what looks like a snake or a beaded necklace is a protein. Eventually that protein gets created, which again we'll have another lecture on how that happens, and then it gets packaged up in these little vesicles, which then moves over to the Golgi apparatus. Here you can see the Golgi apparatus. It is finalizing its packaging here of the protein, and it is now sending it out of the cell. So again, it goes from the endoplasmic reticulum rough to the Golgi for finalization, and then from there the Golgi sends it out. Just another picture of these vesicles that are being created, and we have a couple different vesicles that we look at both in here and in lab. And the first one is a secretory vesicle, and we call it a secretory because it allows the substances to be released out of the cell, and hence the name is secretory. It secretes it out. On the other end, we could have a vesicle called a lysosome, which is more specific. It contains hydrolytic enzymes, or what we say digestive enzymes, which can break down substances within your cell that may not be wanted. So I'll give you an example right here. Here's the Golgi apparatus, and here it releases a lysosome. So inside here there's these digestive enzymes, and here coming into our cell is something that is unwanted, we'll say. It could be some type of a pathogen or something like that. What the lysosome will do is it will attach to this vesicle, release its digestive enzymes in here, and as you can see, the final product is destroyed completely. So whatever that was, it was neutralized by the lysosome, if you will, and destroyed. And that's the purpose of the lysosome, is to break down anything unwanted. Sometimes these lysosomes can break apart prematurely within the cell and kill off the cell, and so sometimes they refer to these cells as the suicide cells if they do rupture prematurely within the cell. And the last component is the nucleus. It's the control center of the cell. It contains genetic material, DNA, and it has three different regions: the nuclear membrane, which is all the way around the nucleus here, and this nuclear membrane is very porous, as you can see. It allows ribosomes and other things to move back and forth between it, but the DNA should never leave the nucleus, okay? And I will say that not every cell has a nucleus, and some cells have more than one nucleus. In the center, this very dense area right here, is the nucleolus, and this is where the ribosomes are created. And then the DNA itself in a non-dividing cell is known as chromatin, and you'll learn more about that in the lab component as well. We talk extensively about this. Now, in our DNA protein synthesis lecture, we will go through the nucleus a little bit more in detail because that's where we tend to talk about it more. So just to test your knowledge here, which of these is not a function of the plasma membrane? Is it A, it is selectively permeable, B, it speeds up chemical reactions, C, it acts as a site of cell-to-cell interaction and guides cellular actions, or D, it encloses the cell contents? So which of these is not a function? B, it does not speed up chemical reactions. Now enzymes speed up chemical reactions, but not the cell or the plasma membrane, okay? It is selectively permeable. It does have cell-to-cell interaction and guides cellular actions because of carbohydrates, and it does divide and enclose the cell contents. Next question, which structures are rigid finger-like projections that greatly increase the absorbing surface area of the cells? And the answer to that would be B, microvilli. Okay. Now we're going to switch gears a little bit, and we're going to talk about cellular transportation and how things move from one side of the cell to the other. Okay, cellular transportation. There are 4 ways that materials can move from one side of the cell to the other. Now remember, the plasma membrane is semi-permeable, so it doesn't mean that everything will be able to move across from one side to the other, but certainly some things. And there's 4 major ways that things can move through, and then we're going to get a little bit more specific with this. So generally speaking, there is directly through the membrane, through protein channels, and then through protein carriers, and then finally through vesicles. So let's just look at these 4 general ways first, and then we'll get a little bit more specific. Directly through. Well, substances can move that are very lipid friendly, meaning fatty friendly, can move and dissolve right through the lipid bilayer. It's like they just melt right through the butter, if you will. So some examples of this would be our gases of oxygen and carbon dioxide. That's a good thing because we know our cells need oxygen, and we know we need to get rid of the carbon dioxide. So this allows these to move easily from one side to the other. They just melt right through. Anything that's lipid or fat-based, steroids, cholesterols, things like that, they can move in and out of the cell. And so water, though, as an example, is not lipid friendly. You know, oils and water, separate, if you will. So water doesn't go directly through the plasma membrane. So again, it's very specific as to what can directly go through. Water would have to go through one of these, basically a channel, if you will. A protein channel is a protein that has a little channel that runs through it. It is determined by the size, shape, and charge of the channel. And it's very specific. So sodium has its own sodium channel, and you can see potassium chloride has its own. Water would have its own. They call them aquaporin channels, if you will. In fact, if I go back, I'll just show you. If we go back to this picture right here, this is a channel right here. And so you can see that, things can pass directly through, but again, it's very specific at the same time. Now, a carrier, which is our next one that we're going to cover, is similar to a channel. Let me just go back here. Similar to a channel, it is a protein, but it doesn't have a channel that goes through. It's kind of like a little puzzle piece, if you will. Whatever it is fits into it, and then it kind of shuttles it across to the other side. So it's not a channel, but it is very specific. It is a protein. They don't really give you a good picture on this last slide that I showed you here, but like these larger ones that go all the way through here, these would be basically carriers. And what would happen would be you'd have a structure, let's say sodium molecule or some type of a protein or something out here. It would attach to this, and then it would shuttle it across in a carrier format. So it doesn't have a channel like our last one. So it's a little different. Go back here. And then the last way is via vesicles. And so our vesicles can move in and out of the cell. They can carry very large non-lipid molecules, smaller pieces, whole pieces across. This is an active process, meaning it uses energy. And so that's something it's probably good to know now, that the word active means it utilizes ATP or energy. And so the two different ways that vesicles can move through are exocytosis and endocytosis. So exo means moving it out of the cell. Endocytosis means moving it into the cell. And I will say that there are only one way of exocytosis that we'll talk about, and 3 different endocytoses. So it's not just as easy as exo and endo. You know, there's different ways of endocytosis, and we'll look at that in a little bit. So now we're going to get a little bit more specific here. This is looking at passive transport. So active transport uses energy, passive transport does not use energy. And so we have diffusion, osmosis, and filtration. And as you can see here, diffusion has 2 different forms. We have simple diffusion, and we have facilitated diffusion. And we'll look at what are the differences between them. So the law of diffusion states that substances will move from one side of the cell to the other as long as there's a concentration difference. So meaning that you have a high concentration on one side and a low on the other. Now it will always move from a higher concentration to a lower concentration. It can never move from low to high under passive transport. Now that's, if we used energy, we could definitely push from low to high because we're using some energy to do that. But in this case, it's passive. It doesn't use energy. It's going to go from high to low. So if you had 100 molecules of sodium on one side of the cell and 25 on the other, they're going to move from that 100 to the 25, okay? And that's your diffusion. There's no energy required for this, as you know, but the rate of diffusion can be influenced by a few things. So the first thing would be the concentration gradient. And so what I mean by that is the more you have on one side, the faster they're going to move. Temperature. The hotter the solution is, the faster they will move. Size of molecules. The smaller they are, the faster they move. Viscosity looks at how thick the substance, the fluid that they are in, okay? The thinner the fluid, the more watery, if you will, the faster they will move, okay? So now you know the opposites, right? Then if they're not as concentrated, they move slow. If the temperature is cooler, they move slow. If the molecules are bigger, they move slow. If the substance that they're sitting in, the solution, is very thick viscosity, they move slow. The final product is equilibrium here. You know, if we have X amount of solutes on one side and X amount of solutes on the other, eventually they're going to equal out, so both sides have about, that's the key, about equal amounts of substances, okay, because it's not going to be perfect ever, okay. An example of this would be a sugar cube in coffee. You put a sugar cube in the coffee, eventually the sugar dissolves in the coffee and it diffuses from an area of higher to an area of low, and they should be equal. I'll show you examples of this in just a minute. So here you can see this animated slide. You can see the substances moving from one side over to the other, and then they equal out, or about equal out. Here's a non-biological example of diffusion. You put a sugar tablet in a beaker of water or coffee, whatever, and the sugar dissolves, as you can see, and notice how the sugar is moving to where there is less sugar, and eventually the beaker is equally concentrated with sugar. So if I took a sample from the top and the bottom, they should be about equal, okay, and that's the diffusion idea here. Now, diffusion, simple diffusion, in a cell moves directly through the plasma membrane, where facilitated requires a protein carrier channel. And it's still passive, though. It doesn't require ATP. It does go from a high concentration to a low, as before, okay, but it has to use a carrier. So let's take a look at this right here. This is simple diffusion. So here's all of our substances out here. Here's a little bit down in here. They move from high to low directly through the plasma membrane. And facilitated diffusion, as you can see here with the other ones, they have to use a carrier or a channel. And here's a better picture of a carrier I was trying to explain to you, like whatever it is fits perfectly in here and then it kind of shuttles it across. But this is facilitated diffusion here. And so it has to use a channel or a carrier to move from high, again that's the key, high to low here. Here's water. Water molecules going through an aquaporin, and that's shuttling them across to the other side. So if these two sides are equal, we're not getting any movement at all. And that's the idea, though, is that this side and this side are going to equal out in the very end. That's the end result of diffusion, is equilibrium. Next we have osmosis, which is basically water moving from an area of high to an area of low. But there is some subtle differences with this. Because we do have solutes that are in the water do play a role in this, okay? So where there is less solutes in the solution, there means that there is more water. Where there is high solutes, there is less water. So water is still going to move from high water to low water, or more water to less water. But there is also the solute thing that's in there as well. And solutes are basically anything that are dissolved within the fluid, so that could include any of the following that you see here. I guess apparently I thought potassium was pretty popular. I put it in there twice. But here is an example of osmosis here. And osmosis, you can see over here on the left, this is a beaker of water, and water is in blue. In the red are the solutes, and there is a semi-permeable membrane running down it, and you can see on the right side there's a lot more water but less solutes. Over here there's less water and more solutes, so the water is going to go from this high water to the less water, or the more water to the less water, and when that happens the beaker fluid on this side goes down because most of it went over here, and then that goes up. Osmolarity is the total concentration of those solutes in the solution, and this osmolarity can change the concentration of the solutes. This osmolarity, the total concentration of solutes in the solution, can change the tonicity of the cell, and the tonicity of the cell is the shape or tone of the cell. And we can change the shape or the tone of the cell by altering the internal volume of the cell, meaning water. So solutes within the cell do play a role in whether or not the cell has enough water or has less water. And so osmolarity of a solution dictates the tonicity. So these solutes right here inside the cell will dictate the tonicity. And I'll explain that here in just a moment. We're going to cut away and go to a separate video. Okay, here we're talking about solutions, and the solution is what the cell is bathed in. It's the watery substance around the cell. It's not inside the cell. And so when we talk about solutions, we're really concentrating on what's the conditions around the cell? What is the solutions around the cell? The inside of the cell does play a role in that, but mainly we're concentrating on the outside. What we have here is the X equals solutes. Remember, solutes can be anything dissolved within fluid. Our blue is going to be our water itself. So we'll first start by looking at isotonic solution. And as you can see here, we have the X's here, solutes, are equal on both sides, and because they're equal, the water is equal as well. Okay, so this solution out here is equal to the solution on the inside of the cell. Because that's the case, we shouldn't see movement of water in dramatic fashion. We see a little bit of water shifting in and out, but not dramatically. Okay, so this is going to be homeostasis right here. And this is what we would desire our cells to be in, is this isotonic solution, because the solution on the outside is equal to the solution on the inside, and we just have a little bit of fluid moving back and forth. Now, That does change if we go to a hypotonic solution. So remember, we're looking out here. Hypo means less, and we're looking at less solutes up here, okay? So because there are less solutes, we have more water, okay? Remember, if we have less solutes, this equals more water. And in this case, you can see that there's going to be more water out here because there's lesser solutes than on the inside. So we should see water molecules moving from high to low concentration gradient. Now when this happens, this is very extreme. This is under not normal circumstances, okay? So if you were to overhydrate yourself, which can happen, the water can shift from the outside of your cells in, because that's the only place it can actually go. Now when we see that in dramatic fashion, when we see this water shifting into the cell very rapidly during this hypotonic solution, the cell begins to swell up and eventually it can rupture, and this is called lysis when it does so. So here's an example that I can give you. About 5 or 6 years ago in California, there was a couple radio stations that the disc jockeys decided to play a little game. And they said, who can hold their pee for a Wii, a Nintendo Wii? So that's when the Nintendo Wiis were really popular. They had people come in and they had them drink excessive amounts of water and see who could actually hold their pee the longest, and whoever held their pee the longest would win a Nintendo Wii. Well, the problem was that these people drank so much water, the water, when they digested it, it went into their bloodstream, and then from their bloodstream, it went in between their cells, and then in between their cells, it had nowhere else to go but into their cells, because they had so much water on the outside, that it started to enlarge their cells, and eventually their cells started to rupture. And the cells that were rupturing were their nervous system cells. And as you know, our nervous system cells we can't get back if they are injured. And so actually 2 people died because of this. This is water intoxication. And even though we know that water is really good for us, anything in large or too much or too less quantities can be dangerous to our life. And that's exactly what happened here. So 2 people died in that situation. So they just had water toxicity, they had water overload here. The water has nowhere else to go but into the cell, which causes it to build up and then rupture. Just think about like filling up a water balloon. Eventually that balloon's going to rupture when there's too much water in there. On the other end of it, we have hypertonic solution. So here, hyper means excessive solutes. Okay, so here we have the opposite. We have a lot of solutes, which equals less water. Okay, so because there's more water in the cell here than on the outside, the water wants to shift from high to low. So we get water that moves from one side of the cell towards the outside. What happens, and again this is very dramatic, it's very fast, when this happens, the cell shrivels up like this, and we call that the cell has crenate or crenation. And basically this is essentially dehydration. So we had overhydration here, the water shifted in, and over here we have dehydration where the water shifts out. This is because of the solution on the outside, okay? So you think about this, you know, in your bloodstream here, you're sweating, okay? So you're losing all of this water in your bloodstream to sweating, okay? The water then here has to move into your blood to compensate for that. And then the water here now has a lot of water, so it shifts out. And then in this situation, the cell is ultimately the victim of dehydration here, of the sweating. So this is a hypertonic solution. So ideally, we would like our cells to be in this isotonic solution, but in dramatic situations, we could see hypotonic or hypertonic. Obviously, we don't see a lot of water intoxication. We do see, though, a lot of dehydration, more so than anything else. So these are your 3 solutions, and we'll be talking more about those on the lecture. So as you saw with the video, we have three different solutions. We had the isotonic, which was the solution has the same concentration of solutes on one side versus the other, which means that the water is fairly equal, and this is homeostasis, so we didn't see much movement. Hypotonic is where the solution had lower concentration of solutes than another, meaning that it had more water outside, which means that the cell would attract that water and potentially enlarge and swell or burst, which is called lysis or hemolysis if looking at a red blood cell. And we also saw when a solution is hypertonic, which means that it has a higher concentration of solutes than another, meaning it has less water than inside the cell, and therefore the water on the inside of the cell moved out, causing crenation or shrinking of the cell, which is what we saw in dehydration. And here just summarizes those different solutions again, isotonic being homeostatic and homeostasis, and we have hypertonic where the water leaves the cell and crenates it, shrinks it, and hypotonic where we have too much water on the outside and the water shifts in, swells the cell, and potentially that cell could lysis. Filtration is another way we can move substances. This is moving substances from a high pressure to a low pressure. And so really what we're looking at high pressure being high pressure of water, such as hydrostatic pressure is what we call it. But filtration again is moving something from a high pressure to a low pressure. In the kidneys, we filter things out of our blood in the kidneys, and that's because of a high pressure to a low pressure. In our capillaries, in our small little blood vessels in our body, we move substances out in between the cells because of a high pressure to a low pressure. Small particles in water move very easily where large molecules will not pass, they're too big. This is still a passive process, meaning it doesn't require energy in order for that to happen. You can just see here as an example, in a capillary we have blood flowing through it. This pressure that's coming down pushes substances out, and that's your hydrostatic pressure here. That's all I want you to really look at with this slide. Okay, so those are your three passive transports. That would be diffusion, osmosis, and filtration. Those three do not require energy to move substances, whereas now we're going to shift gears into active transport, which do require energy to transport substances. They require protein carriers to be utilized. They can move substances from high to low, but they can also, because we're using energy, move materials from low to high. Some active transports can exchange one substance for another, which we're going to look at right now. Our first one is called a sodium-potassium pump. And what this is doing is it's moving sodium and potassium in and out of the cell. It's an exchange process. So here you can see this is just one continuous sodium-potassium pump here. So they're just taking you step through step here. So this is a sodium-potassium pump. Notice it is a carrier here in the plasma membrane. Here's the sodium, and you can see the sodium are moving into the pump. Here comes our friend the ATP that's needed. It uses that energy. It pumps out the sodium. At the same time, this potassium in yellow here is working its way into the pump, and that gets pumped into the cell. So we're exchanging sodium, in this example, out of the cell with potassium moving into the cell, and it has to require ATP in order to do that, along with a carrier or a channel to do that as well. In this case, this is a carrier. So this is an exchange process. So the big difference between passive and active, again, is passive doesn't require any energy to move things back and forth, but our active transport does. I did put in a couple of videos here. You can kind of watch those on your own. I'm not going to take the time to review those at this point. I don't think it's going to be, you're going to learn anything more right at this point at least from that, but I would encourage you to look at those on your own. And the final ones of other forms of active transport are what we call vesicular transport. and our two vesicular transports are endocytosis and exocytosis. And so here we have three different ways to get substances into the cell called endocytosis, and they are receptor-mediated endocytosis, phagocytosis, and pinocytosis, and only one way to get rid of something called exocytosis. So when we look at endocytosis, bringing things into the cell in the formation of a vesicle, I want you to understand that a vesicle is a membrane-bound sac, and basically it's like a small plasma membrane sac, basically. And this does require energy to move things back and forth via endocytosis. And the first one that we'll look at is the receptor-mediated endocytosis. And this one is very specific because it has very specific receptor sites. And I'm going to go up one slide here so you can see that. So this is the plasma membrane. And on here in purple, these are the receptor sites. And these little green structures here are what we call ligands. And ligands are specific target molecules that bind to those receptor sites. So as you can see here, these ligands in green here, it's just a general word for a specific molecule. Okay, that's all ligands is. So the ligands, they bond to these receptor sites here, and this starts to form a vesicle, and eventually that vesicle moves into the cell. This is why it's called receptor-mediated endocytosis. And you can see that. Now, if there was triangles out here, let's just do the shape thing. If there were triangles out here, the triangles would not be able to fit in these receptors. So these receptors are very specific to this molecule right here. And again, we call these molecules ligands. They could be anything. I just gave you a couple examples here. They could be hormones or proteins, but you get the idea. Phagocytosis is cell eating. They don't require any type of receptor sites, but these are cell eating. They take in very big, large, solid particles, as you can see in this picture here. Here's our cell. It starts to form a vesicle around this particle and eventually brings it all the way in the form of a vesicle. Now, they could be anything from large things such as bacteria, cell debris from dead cells, or even foreign particles. These things have even been known to try to gobble up like shreds of glass that might be in your skin from an injury, but obviously they can't do that, but they attempt to do some of that. What we see here is it then moves over into this, and this is where the lysosome we were talking about earlier comes in and breaks down that substance and eliminates it. So we kind of talked about that earlier. Again, this is active transport, vesicular transport, bringing something in via vesicle. Here it is. Now you notice there are the receptor sites here, but we're not using them, not for phagocytosis. This is just the cell-eating portion of it, taking in the whole product. Pinocytosis is what we call cell drinking because it takes in smaller molecules with the dissolved liquids within it. So it does not use receptor sites. It's taking in smaller substances, as you can see, into the form of a vesicle. Here you can see it transporting substances from the blood all the way across to the other side of a cell. So that's the last of the endocytosis. So we had receptor-mediated, we had phagocytosis, and pinocytosis. And now the last thing is exocytosis. This is taking something in the cell and removing it via exocytosis. So here you can see the secretory vesicle with substances in it. It latches onto your plasma membrane and allows that substance to release out. Examples of this would be your digestive enzymes within your intestinal tract, your pancreas, your salivary glands. This is how your saliva is released in your mouth. Mammary glands for females that are pregnant and breastfeeding. This could be breast milk here in the breast, and this is how the breast milk is released as well. And you can see that in action here for the most part in a very high magnified photomicrograph of exocytosis here. And you can see that substance leaving. Here's that vesicle. Now what happens to this vesicle right here? This eventually just kind of works its way up and becomes part of the plasma membrane. Okay. So this is the cell and the cell transportation part. You do have a few PowerPoints and lectures in this unit. If you have any questions, feel free to give me an email.